Yeah, you're good.
Okay. All right. So on Wednesday, we had just started our discussion about passive transport. So remember, the key behind passive transport is that we want to move from an area of high concentration to an area of low concentration. Okay, so I'm just recreating. Oop, I guess we're staying childlike. That's fine. I don't care. Today, I don't care. Okay, so we want to move from having no elbow room to an area where we have all the same amount, right? AKA equilibrium. Okay. So remember that that's true for all types of passive transport. Okay. And so the thing that sets diffusion specifically apart, and so what we're going to find is for passive transport, we have 3 different ways to do this. Okay, so remember, the goal here is we want to get things across the membrane and into the cell, like food, water, air. Okay, but the membrane is still supposed to be keeping the bad stuff out. Okay. So we need several different methods that allow us to keep the good stuff in while protecting ourselves. Diffusion is our first method. And this is the simplest. Okay, simplest meaning least complex, right? Not easiest. Okay, so remember, if we consider, there's our phospholipid bilayer. Right, we have our phosphate heads, our little lipid tails. Everything is really close together. Okay, so simple diffusion takes advantage of this fact. And so it's going to utilize the power of very small molecules, okay, something like oxygen or carbon dioxide. Okay, so things that are very, very tiny, and allows them to slip just between our phospholipids. Okay, they shimmy between. Okay, so we're still following all of these other ones. We're moving down gradient to equilibrium. Okay, but for diffusion specifically, okay, we can only be teeny weeny, okay, because we're gonna shimmy. Okay, we're not going to use any other method besides we're going to squeeze our way through. And so small things can do that, but remember, really, really big things, possibly the worst sugar molecule I've ever drawn. Okay, if we consider something like our really poorly drawn glucose molecule, if I wanted to try to squeeze through, okay, I'm going to be too chunky to fit. Okay, so this is one of the keys, and this is going to set us up both for why only our small molecules will work and why we have more than one kind of passive transport, right? Because we need things like glucose molecules in our cell, so we need a way to get them in, hence things like step 2 and step 3. Right? How do we actually get them in when you're too big to squeeze? Okay, but diffusion works fine for something like air. I think this is where we left off on Wednesday, but I wanted to make sure we sort of retouched base on it. Does this feel okay? Okay. So diffusion is like a very exclusive club, right? Tiny molecules only. But we still have to fulfill all of the rules of passive transport, right? Moving from high to low, right, and moving towards equilibrium. Okay. Remember, at equilibrium, okay, solutes, okay, whatever it is that we're using, okay, oxygen, carbon dioxide, eventually things like glucose, everything's still moving. Now, we talked a bit about how this is like kindergartners moving seats around. Okay, everybody's still moving around. Just like the name implies, we're moving back and forth so things are equal still on both sides. Okay, the key thing to remember, okay, if a cell isn't moving, it's dead. Okay, just like if you're not moving, you're dead. Okay, even when you're sleeping, you're still moving, right? You're breathing, you're tossing and turning, okay? Your eyes flutter, okay? You're still moving. Your blood is still pumping. Okay, and the same has to be for the cell. There still has to be movement inside it. Okay, so even at equilibrium, we still have to have some movement. And we're just not having any change, right? It's just the annoying kindergartner. Ah, it's my Jenny! Okay, passive transport and then diffusion itself feel okay? Okay, so this looks a lot like the drawing that we had on our last page. Okay, just cleaner. So if we start on the far left, right, I have an area of high concentration. On my left side, okay, and low concentration on my right side. Okay, and our orange molecules want, okay, to spread out. We want that elbow room. Okay, so in this case, we're a small molecule. We can use diffusion. So we're able to cross that membrane. Okay, shimmy. Okay, until we spread out evenly. Equilibrium is equal. So we have about the same number on both sides, okay? But that doesn't mean that we stop or we're dead, right? So we can still see a little bit of exchange going on, okay? I want to sit by Jenny. Fine, I want to sit by Hank. Right? So there's still a little bit of movement, okay? But it's not all in one direction anymore. And that's what we mean by equilibrium, is it's equal exchange now.
So somebody asked in the chat, how are you sure if there is no movement at equilibrium? Like, how can you tell?
You mean how could you tell if the cell was dead?
I think so.
Okay. So in this case, so I guess I'm not sure I understand the question. Can you rephrase it for me?
she said if the cell is dead, I think she said like if the cell is dead, nothing would be able to move.
Correct.
Yeah.
Yes. Yes. So if the cell is dead, nothing would be able to move. So in that case, and so I have, so we'll see this a little bit later on. And so what you will find is not only would these things not be exchanging, but whatever is inside the cell, eventually things would also get used up. Right. And so while we see things occur at equilibrium, if this is air, right, oxygen, this is far more advanced than you guys will need to know. But this stuff is also getting used, right? If this was glucose, we would be eating it, breaking it down and using it for things. So equilibrium is states that we're working towards, but we don't stay in this state permanently, right? Because the cells are using these things. When you breathe in air, it gets used in your body. So all of the cells are in a constant state of change. So if the cell was dead, right, not only would these particles not be moving, but they also would not be used. We'll have a lot of good examples of that as well when we get into metabolism of dead cells. I hope that helps. Waiting for the click. Okay, we'll back up if I hear another click. Okay, so the other thing to remember, I'm going to clear this off because that looks weird up there, is when we're looking at diffusion of anything, the phrase I like to use is solutes are selfish. Okay, and what I mean by this is that each solute or each different thing that is going to be diffusing across only cares about itself. Okay, it doesn't care about anything else that's in there. Okay, so when we're talking about areas of high concentration, it only cares about its own concentration gradient. So if we look at the example that I have, okay, the orange particle, okay, is only going to follow its own concentration gradient. So it has an area of high concentration on the left. And our purple particle has its own concentration gradient, entirely separate and independent of the orange particles. Okay. So it has a high concentration gradient on the right. And this is super important if you think about it from an applicable standpoint, right? If I need oxygen, that oxygen needs to follow its own concentration gradient, okay? So I can breathe regardless if I'm full on food or not, right? So I can't have the idea that I have lots and lots of glucose inside my cell stop oxygen from coming in, right? Even if I'm low on oxygen. So all of these particles move independent from each other. So when you're doing a diffusion problem, treat each part of it totally separate, okay? So you can kind of do it in a step-by-step process. So the first thing I'm going to do is look at this and go, I can't really do this on the, I don't know how to do this on the, okay, what is the orange stuff going to do? Okay, I'm just going to cover up all of the purple stuff and say, okay, what are my orange particles going to do? Okay, I picked up my pen. Oh, come on, man. Okay, so my orange stuff is concentrated on the left-hand side. It wants to flow down its concentration gradient to where it is less concentrated. Right? So my orange stuff is going to go from high to low. until it reaches equilibrium. Okay, then I'm going to go all the way back to the beginning and say, okay, what is my purple stuff going to do? And ignore everything I just said about the orange stuff because none of it matters now. And say, okay, well, my purple stuff is high on the right side, right? So that stuff's going to move high to low. So it can get elbow room. And it's going to do that until it reaches equilibrium. An equilibrium that is entirely independent of whatever the orange guys are doing. It's like they don't see each other. Okay? It's like parallel universes. Okay, this works great if you're a sci-fi geek, like me. Okay, so take these step by step. First say, okay, what's this one going to do? Solve that part. Okay, good. Now what's this next one going to do? Solve that part. Okay, because they never interact. They don't do anything with each other. Okay, that feel good? Any questions about that? Okay, so your mantra, solutes are selfish. Okay, so we're still in passive transport now, but remember we talked about what do we do if we want to get something that's bigger across our membrane. Okay, so here again we have that glucose that before was too big to shimmy across. Okay, no shimmy. Okay, so unlike oxygen or carbon dioxide, okay, that could sneak across the plasma membrane, okay, our glucose or sucrose or whatever it might be, okay, needs a different way to go about it. And that's where facilitated diffusion comes in, right? Facilitated literally means to help. Okay? The help in this case comes about in the form of our favorite biomolecule, proteins. Okay? Proteins in this case embed themselves on our phospholipid bilayer, our plasma membrane, and act like tunnels or gates. These tunnels and gates, okay, are much larger openings. Okay? They still act like security guards, and so they won't just allow anything through. Okay? But they're big enough to allow good, delicious, important things to be able to pass across now. Okay? Oxygen and carbon dioxide can use these too, and they can get across much faster than squeezing themselves across the membrane. Okay, but for the most part, our chonkers are using this. Now, despite the fact that we're using some pretty fancy looking gear, remember that this is still passive transport. Okay, so this is still free. Remember, ATP is that currency form. So pretty much any time that we're moving from a high concentration to a low concentration, so we're still moving down gradient it's still going to be free of charge. Even though we're using a gate and a security guard, okay, a free service. Okay, so our membrane proteins, okay, are allowing things that are bigger to cross, right? And really anything bigger than those teeny weeny molecules. Any questions about that? Well, that's kind of a big topic. So unfortunately, I think that's going to be it.
Did somebody ask in the chat if this stuff will be on the test? And just to make sure y'all know, whatever we have covered from the beginning of class until today is fair game for the test. And I know that's not the case.
Well, you answered it in the chat box though.
Yes.
Yeah, you got it covered. You may find that for your review quiz, there may be a few questions that go further than this, that cover active transport. Those will not be on the test, but I can't go back and fix the review quiz now, right? Because I didn't know how far we were going to get today. So it's fine to try those. We'll cover those when we start new material, but don't be upset. You covered them in lab, but just because we did them in lab does not mean that they will be on the lecture material, right? So we will only go up to facilitated diffusion on your lecture exam. Okay. Once you are done, back 2 rows, you are free to go. If you have questions, guys, do make sure that you get help, okay? Remember, it's better to get help than sit frustrated. Oh.